So now it's time to talk about neurology. Neurology is a really complicated science, and it's based on little cells called neurons. The neuron is the basic functioning unit of an animal nervous system. Plants don't have nervous systems, just animals do, and more complex animals have more complex nervous systems than the simpler ones. A neuron is a weird cell. It has a roundish area. All neurons have a roundish area that is the cell body. The cell body actually contains the nucleus, which is responsible for transcribing, RNA, and then there's lots of ribosomes and ER for making proteins. There's a bunch of little short fibers attached to the cell body, and then there's usually this one long fiber that comes off, and then it branches with little short branches at the end. The little short things attached to the cell body, those are generally all called dendrites. They pass impulses to the cell body. Okay? The axon transmits impulses away from the cell body to the axon terminal. So general impulse flow goes from dendrite to cell body down the axon to the terminal. Now the axon, and sometimes dendrites, it depends on the neuron, they're covered with these yellowish orange cells. Those are called Schwann cells. Schwann cells are made of myelin. The Schwann cells actually, it's a big, long, rectangular cell, and it's rolled up like toilet paper on a roll around the axon. And each Schwann cell leaves a little gap between it and the next Schwann cell or the adjacent Schwann cell. What that does is causes the impulse to jump from node to node as it moves down the neuron. And the jumping is actually much faster than just flowing. Impulses often flow down dendrites, flow across the cell body, jump down the axon to the terminals, and then the neuron releases what are called neurotransmitters to stimulate the next neuron. Now it's time to talk about action potentials and resting potentials. So let's review for a second the sodium-potassium pump. The sodium-potassium pump is responsible for the resting potential of a neuron. And it's based on this yellow thing here in the middle. What this pump does, is it pumps sodium out. There's the sodium, and it pumps potassium in. So the dark blue balls are sodium, and the light blue balls are potassium. And so what's happening here is it loads, and then ATP energy is used, and it throws the sodium out, and it throws the potassium in. And it's a 3-to-2 ratio. For every 3 moles of sodium pumped out in one cycle, 2 moles are pumped in. So the sodium-potassium pump works per cycle, 3 ions of sodium being pumped out to 2 ions of potassium being pumped in, and the consumption of 1 ATP high-energy phosphate. Then the green thing here that lets the sodium flow back in, that's a sodium-gated channel. That's what is the essence or the foundation of a nerve impulse, the actual impulse. The pump sets the stage for the impulse, but the green thing is the actual impulse architecture. The purple thing here lets the potassium flow back out to help recharge the neuron so that the neuron can fire another action potential later on. So let's go back now to the unit we were on and talk about communication. Let's see if I can get this to go back. Let's see. Okay, good. Now let's go back here. All right, good. Now, it turns out, so we just talked about all the sodium-potassium pumping. 3 sodium ions are pumped out while 2 potassium ions are pumped in. And what this does is it puts more positive charges outside the cell than inside. As a result, the resting potential of the neuron is about 70 millivolts negative on the inside, positive on the outside. In the end, there's more positive charges outside than inside. On the inside, there are also negatively charged proteins. So the inside ends up, as a result of the sodium outside and the negatively charged stuff inside, the inside's about 70 millivolts negative, and on the flip side, the outside would be 70 millivolts positive. It turns out that disturbances on the neuron surface can disrupt the resting potential and cause what is called an action potential, which is a nerve impulse. What an action potential is, it's a self-propagating wave of depolarization that moves down the axon due to the sequential opening and closing of sodium channels. So let's erase that. And I see that this is not displaying quite the way I want it to. So let's go here and let's shrink this one level, and maybe I can get that. Let's move this back up, and now it is. So the way the action potential works, one more time, action potentials are all or none. They occur maximally or not at all. And what they are is a self-propagating wave of depolarization that moves down the axon due to sequential opening and closing of sodium channels. So basically, down a neuron like an axon, there are just multiple sodium channels all the way down. When sodium rushes in one channel, it trips the next gate open, so sodium will rush in the next channel, which trips the next gate open, and which causes sodium to rush in the next channel, which trips the next gate, sodium comes in, trips the next gate, sodium comes in. It's very fast. Maximum speed at the node jumping down the nodes of Ranvier, it's over 150 to 200 miles per hour. So the inside, during the nerve impulse, it becomes positive because sodium flows in. To recharge the outside of the neuron, potassium flows out, which restores the outside positive charge and the inside negative charge of the membrane. It turns out that myelination on neurons is not just for protection, but also to increase impulse speed, because it turns out that impulses can jump faster than they can flow. So vertebrates in general, animals with a backbone, use myelin on their neurons. Most invertebrates do not have myelinated nervous systems. They use unmyelinated neurons for their movement, but they tend to be small-bodied animals, and their nerve impulses are fast enough to keep them fit. So now that takes us to these two diagrams. This is the diagram of the sodium-potassium pump. We've already talked about that. But this graph's kind of important. This is showing that a resting neuron has a charge of about 70 millivolts. When the neuron is stimulated or disturbed, sufficient to get it to leak a little bit of sodium so that the membrane loses its 70-volt charge. If it drops to about 15 millivolts less than resting, it'll fire a full-blown action potential. And so what will happen is the sodium gates will flip open and then they flip closed. They flip open, the leaking of the potassium back out repolarizes the membrane and flips the sodium gates closed. After that, there's a refractory period where the neuron cannot fire an action potential because it's too disturbed from the previous action potential. But within milliseconds, 2-3 milliseconds, it's back to its resting state and ready to fire the next action potential. So neurons can fire multiple action potentials in a row to transmit information. So now let's see what this actually might look like if you could see it. Neurons that have no myelin have action potentials that flow like this. You see these little plus charges flipping in? Those are the sodium gates. That's the result of the sodium gates opening. Sodium rushes in, potassium rushes out almost immediately to restore the positive charge on the outside. These impulses flow 50 to 100 miles per hour maximum speed. The wider or fatter the neuron, the faster this action potential can flow. On a myelinated neuron, the action potential jumps faster than it can flow. This is vertebrate nervous systems, okay? Invertebrate nervous systems do this. Some of the human body is actually wired this way without myelin, but most of our really important neurons have myelin and transmit impulses by saltatory conduction, it's called. This is saltatory conduction. This is non-saltatory conduction. Saltatory conduction is jumping impulses. Non-saltatory conduction is flowing impulses. Now that brings us to now one of the most amazing parts of the story here, which is synapses. Synapses are chemical space. They're little spaces between the neurons, between the axon terminals of one neuron and the dendrites of the next neuron. And neurons squirt neurotransmitters on one another to relay action potentials from one to another. Basically, what happens is the axon terminal stores neurotransmitter. When an action potential reaches the axon terminal, it releases neurotransmitter. One of the most common in the human body is acetylcholine, which is abbreviated ACH. The acetylcholine is literally exported out of the axon terminal very quickly. It crosses the little space between it and the next neuron, that's called the synapse. It touches a G protein linked receptor on the dendrite, and that causes sodium gates to open, which starts the next action potential on the next neuron. So neurotransmitters are special chemicals, often acetylcholine, that are stored at the ends of axon terminals. The membrane on the axon terminal is called the presynaptic membrane. The membrane on the next neuron is called the postsynaptic membrane. The space between the presynaptic and the postsynaptic membrane is called the synapse. So in this picture over here, we have an axon coming off. There is a synapse right there, and this is the end of the axon terminal, and this is the dendrite. That's the synapse. This is the presynaptic membrane. This is the postsynaptic membrane. The postsynaptic membrane has the G protein linked receptor that receives the neurotransmitter to cause the next action potential on the next neuron. So when an action potential reaches the end here, neurotransmitter squirts across, binds to the receptor, which causes the next neuron to open the sodium gates to cause another action potential. So calcium ions are part of the story here. Calcium ions are in the synaptic knob, which causes some of the neurotransmitter vesicles to discharge their contents. So the reason the neurotransmitter comes out is as the action potential hits the end of the neuron, local calcium release causes some of the vesicles to squirt the neurotransmitters out. The neurotransmitters act as the Pony Express. They cross the membrane and touch the other side. Now, I don't want to give you the idea that all neurotransmitters stimulates, because some neurotransmitters hyperpolarize the postsynaptic membrane, and that inhibits the postsynaptic membrane from sending an action potential. In the human body, probably most synapses have depolarization functions going on, but many synapses, especially in the brain, have hyperpolarization synapses going on. So when you're trying to calm yourself, for example, after a nervous thing has happened to you, like somebody scared you, in the core of your brain, as you try to calm yourself, you try to hyperpolarize neurons to bring your blood pressure down, to inhibit these bad things these extra, hyperactivities that are happening, to slow your heart down, slow your breathing rate down, slow your thought patterns down so you can come back down to a resting state. Like I said, acetylcholine is the most common neurotransmitter in the human nervous system. It basically is very commonly stimulating or depolarizing postsynaptic membranes. When a postsynaptic membrane depolarizes, it will send an action potential down. If it's hyperpolarized, remember, it won't send an action potential down. So this is very fast. Milliseconds after the acetylcholine is released into the synapse, it's broken down by a special enzyme called cholinesterase. If it stays in here, we would become paralyzed and die. So the acetylcholine has to be destroyed as soon as it's used, and then it's recycled back into the axon terminal for more uses. Now, to summarize then, this is how the action potential works. It sweeps down to the end of the terminal, neurotransmitter crosses the gap, and so here we go. Here comes the action potential, neurotransmitter squirts, new action potential, it goes. So one more time, action potential, calcium release, neurotransmitter release, new action potential, down the next neuron. This is a depolarizing connection here where the neurotransmitter is causing the postsynaptic membrane to depolarize. As a result, we have an action potential on the dendrite caused by the axon. Which brings us now to the last part of this chapter. Transmission of information along neurons and synapses is really complicated, and it does result in responses. So the response can be either stimulatory or inhibitory. Excitatory responses in the nervous system include like muscle contraction, another neuron having an action potential, a gland releasing a hormone or something. But inhibitory responses include muscle relaxation, another neuron not firing an action potential, etc., etc. Generally speaking, every time you move a muscle on the opposite side of the joint is inhibited. So while one muscle is active, the other one's being inhibited. In this animation, what we have here is when the hammer strikes the knee. This is supposed to be the quad muscle in a knee. So this is the kneecap right down here. When the hammer strikes the kneecap, the muscle is jerked. At that point, spindle receptors in the muscle fire action potentials. They race down the neuron all the way to the spinal cord, shoot neurotransmitter on the interneuron, which shoots neurotransmitter on this motor neuron, which then carries an impulse back to the muscle to cause it to contract. This is why when the doctor strikes your patellar ligament, your knee jerks out about a half a second later. This is what's going on. So let's see if we can go. I don't know if this is linked. Yeah. So let's erase the screen and let's step through this. Going through this, here comes the hammer now. Let's go here and here. All right. Hammer strikes muscle right there. So let's get the drawing tool going. So the hammer strikes the patellar ligament. Actually, it's the end of the muscle here. That shocks the muscle. That fires the spindle receptor here. Let's go back up here and go, let's erase the page and go to a cursor. All right, now, at that point, that fires the spindle receptor here. That fires a jumping action potential. Notice it's saltatory. It's not flowing, it's jumping. Jump, 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 jump. This is called a sensory neuron. This sensory neuron is carrying impulses into the spinal cord. This is called the cell body. On a sensory neuron, there's a long dendrite and then a really short axon. That's your typical sensory neuron in the human body. And the dendrite has myelin. The axon also has myelin with Schwann cells. So there's the action potential right there. It's jumping, jumping, jumping. It gets to the axon terminal, little calcium release there. Neurotransmitter shoots across, hits the postsynaptic membrane, fires an action potential. Now here it flows. There's no myelin in here. Gets to the end of the axon terminals. Neurotransmitter squirts across to this is called a motor neuron. This is called an interneuron. The motor neuron here receives the action potential, depolarizes, action potential jumps down as it flows out of the spinal cord, down your leg to the quad muscle. In the quad muscle, the neurotransmitter is released. The neurotransmitter touches G protein linked receptors, which leads to the muscle shortening. So this complex interaction of all these parts is made possible by complexity inside and between the cells. The spindle here, the reason it fired is that it was mechanically disrupted by the shock of the muscle. And a muscle that is tight will be less likely to get damaged when it's struck with a ball or a blunt object. So that's why the muscle wants to tighten itself in response to the vibration. When the spindle fires, it sends information into the spinal cord. Here, an interneuron receives the information from this sensory neuron. The sensory neuron relays the information to the interneuron. The interneuron squirts neurotransmitter on the motor neuron. The motor neuron sends the information back out along axon to the muscle, and